Brenda, this is for you. Notice that the title is a question mark because I don't know the answer to the question, so I'm not going to tell you anything that's true. I'm going to be talking about an ongoing project. The ongoing project has ups and downs and discoveries and surprises. And so Sarah and I will be talking about this ongoing project, and actually the next speaker, Mike Turner, is in on this. But we're not going to have a definitive answer for you. We're going to have a whole lot of data that's very, very suggestive. And so the question, can we capture, store, and reproduce healing and get the healer out of the equation? That's really the question. So you think in terms of, well, take a battery. Can we put healing in a battery? And what would store it? And how do we deliver it? And that's the ongoing project. So I've been working on anomalous healing research for quite a bit of time now, and I'm only going to talk about the last bullet. The first, the parameters of anomalous healing, what makes it work better, what makes it work less well. I mean, you have to understand at this point, does healing exist is a stupid question. And anyone who still entertains that, they'll say something like, well, I know you've done all these experiments, but if you could do it in my lab, then I'd believe you. And so would you do the same thing you've done 85 times and do it just one more time and that will convince me? That's not going to convince anybody. People who want to look at data will see that healing exists. What are the parameters? Does belief matter? Does distance matter? Does dose matter? And I can keep going. These are interesting questions, not is healing real? I've looked at physiological correlates using EEGs and functional MRIs, but that's not going to be the focus today. The weak link in all of my studies, of course, is a theoretical model, and Iman Sparus is going to be going in a little while, and he's going to hopefully provide us with some very, very interesting conceptual ideas that can try to make sense of some of these crazy data. But today, we want to talk about practical applications. Can we bottle it? Can we put it in a battery? And if so, what's the carrier? What do we need to make the battery out of? What do we need? How do we bottle it? What are the characteristics of the bottle? Things like that. And if we can put it in a battery, if we can put it in a bottle, then we can make it scalable. So that's the plot. I've done a lot of experiments. I'm not going to talk about even a small percentage of them. These are in vivo experiments with mice. I'm only going to show a couple of mice pictures, but these are in vivo. So I've studied at City University and a whole bunch of experiments, different kinds of cancers in particular, different kinds of cancers in different places, even crazy things like nude mice, extremely aggressive mice. I have a boatload. I just keep going with experiment after experiment of in vitro data, which Sarah will be talking a little bit about now. But I come with a reasonably long history of research and a boatload of data points. And I can demonstrate, the first one isn't particularly interesting, anomalous healing. Does it happen? Yes. Time to move on. It's not an interesting question. I can demonstrate, though, that there's a dose response. And so I don't know exactly what the dose should be in every indication, but I can demonstrate, it's not the focus for today, that there is a dose response. It's not healing, hocus pocus or magical thinking kind of stuff. Something is being delivered. And something is being delivered not all at once. I don't think it's energy, even though people talk about energy healing. I consider it more information, but that's another talk. But there is a dose response. If you want to talk to me on the side, I can show you how I came to that conclusion. I can also demonstrate that healing is not a linear response, that it occurs rather in phase transitions, that you don't have something like something gets a little better, a little better, a little better, a little better. Rather, healing, sometimes all symptomatically, at least it gets worse, and then suddenly it reverses. And so it's almost as if the condition collapses. And we see this in humans, we see this in mice, we see this in cell cultures, we see this all over the place. Healing is a non-linear response, and it also has methodological implications in the sense that if I do an experiment, and let's say I've got a bunch of mice and I give them whatever I give them, and we're going along, and I stop the experiment at the end of a week, and I'd say, well, it failed. But if I go 2 weeks, it's dramatically different. And so there's all sorts of methodological implications from nonlinearity. I'm reasonably confident at this point, but not the focus of this talk, that healing is information rather than energy. Out there in the world, people talk about energy healing. I don't think there's any energy. I think the energy is in the healee, not in the healer, and I don't think any energy is transmitted. I can demonstrate, but not today, that there's a resonant bond between healer and healee that's fluid. I can show phase locking of brains at a distance using EEGs, using functional MRIs, using something links physically separate groups together. Whether it's human to human, human to mouse, cell to cell, something bonds them together. Not the focus of this talk. I can also demonstrate, not the focus of this talk, that healing is not due to a particular state of mind on the part of the healer. Belief, I suspect, probably retards healing. Belief on the part of the healer retards healing. Belief on the part of the healee retards healing. It's pretty interesting. Healing is rather an autonomic response to need. And I'm going to, a couple of slides that might indicate that, but it would be a talk unto itself. And I could keep going on and on and on. So for today, the question is, can we put it in a bottle? Can we stick it in a battery? Can we make an app? A healer app, a killer app, I don't know, whatever you want to call it, who knows. So just a couple of pictures. The SSE folks have seen hundreds and hundreds of mice studies and slides, and they're sick of it. For the newbies, just a couple of slides here. The basic model that I use is I take something off the shelf, meaning an already proven existing model, a mouse model. In this particular mouse model, there's more than 2,000 published experiments. You know what's going to happen to them. And I introduce a single variable. What happens if I put healing into the equation? What changes? I mean, that's basically the model. Whether you're dealing with cell cultures or you're dealing with mice, what happens if I interject healing into the model? In this particular one, we got a bunch of mice that die within a month. And this is probably the most commonly used experiment, or the most commonly published experiment. It's ugly research in non-metastatic cancer. You inject a mouse and the tumor gets bigger and bigger and bigger. You don't need to be a biologist. One leg is gone inside the tumor. The fur is falling out. The eyes are dull. This is essentially a dead mouse. And we introduce into this off-the-shelf model the variable of healing. So in particular, we try to get really, really good-looking people to put their hands around a cage. One of our mysteries is that ugly people can't heal. We don't know what that's about. I'm trying to get a grant for that, and I think we'll find out tonight how to get that. So why can't ugly people heal? So we introduce healing, and healing is extraordinarily boring, and you treat cage after cage after cage after cage. In this particular experiment, there are 6 rooms going on. I just don't out anybody else. So there's other faculty members in different rooms doing the same thing. There's students in there, and they're doing the technique that I developed. And you do that, and then you get pretty interesting results. The tumors grow, and then the tumors start to develop a blackened area, and then the tumors ulcerate, and the ulceration can be really ugly. You'd think that was a sick mouse. Turns out that's almost a cured mouse, and very soon after this, you're going to have a phase transition, and the thing is just going to heal. So we deal with a model that has 100% death, and we get 100% cure. You don't need to do stats, says the statistician. And I give you a quick illustration of a phase transition. This is a particular mouse. It's a different mouse in the last picture. But you see, the ulceration is not as big. This is 22 days subsequent to injection. I'll show you the same mouse 6 days later. That's a phase transition. And so we're not talking about, I'm getting a little better. I've got cancer. Histology will show cancer in that, and then it's gone. And so if I had stopped this a couple of days earlier, then I would say, no, the healing didn't work. But if you carry it out long enough, suddenly there's going to be a transition. And so once again, because it's really cool to look at, here I am 6 days later, and that's the biologist pulling back the fur looking for where it was. If you don't pull back the fur, and there's no sign of cancer, and the mouse will live its entire lifespan. More than that, it's immune to further injections for its entire life. You can do it with people, but you have to deal with very small people because they don't fit in a cage necessarily. And they squeal. I don't know. We don't know what that's about. Again, I'm seeking a grant for that. But you'll see stuff you don't want to see, and then suddenly you'll see stuff that you do want to see. You can do this by thermal imaging. You can do this by cancer markers. You can do this in a whole variety of different ways. So the question is, can we bottle this? What stores healing, and what can we use? I think a battery is a better analogy. So can we use this and stick this in a battery? And can we do this in biology, or should we take a biological approach to this, or should we take a physical approach to this? What's the best way to make a battery? So the first thing we got to do is we see if can healing be stored, and what can it be stored in? And it turns out it seems to be able to be stored both in biology and as well as in stuff. We'll show you that in a second. So we'll just do the highlighted ones. I've already said mice are immune to repeated reinjections of cancer once they're cured. Now, this next bullet is reasonably interesting. An infected mouse can be cured by cell transplantation from a treated mouse. This, I'm saying, is a storage facility. This is biological storage. And just as a side thing, we've killed some cell lines. You get a mouse, it's got a tumor, you grind it up, you seed it, it moves forward. Well, we kill the cell line. But that means something happened, something biologically is going on. So, stupid little cartoon here, you got a mouse on the left that has been treated with the healing, and it's got an ulceration, and you take some cells out, and you stick it into another mouse without further treatment of the healing, and everybody's cured. Something's in the mouse, and that's what I mean by biological storage. Now, what if we treat stuff? Well, this is a particular experiment done at UC San Diego, where we treat stuff, the nutrient in which cells are grown. And so the question is, can we store healing in the medium? So we treat that, we ship it off to a lab. And I'll give you another example of phase transition. This is a weak, we're just going to do this crudely. Over here is controls, and here are stuff that's been grown in the treated medium at full and half dilution. And what we have here is after 1 week, nothing happens. If you take a mouse earlier and you look at it, nothing happens, but suddenly everything changes. At the end of 1 week, nothing happens. There's no change in growth, there's no change in necrosis, and then suddenly in the second week it explodes. In this particular case, the treated medium acts as a contributor to the acceleration of growth. So cancer cells thrive in treated medium. Notice, please, healing is not killing. And so cancer cells, as cancer cells, is the client of the healer. If they're exposed to healing, then they explode, they thrive. We haven't done this yet. If we stick it into a mouse, I'm predicting it'll collapse. I'm almost always wrong, Brenda, so don't write that down. Yeah. But we got to find out because, hell, I don't know. If we treat water bottles, so the previous was organic, this is not organic. We treat water bottles, and this, of course, is Jerry's turf. If we treat water bottles and we treat the water bottles, we feed them to mice we never meet, those mice get cured too. It would appear healing is being stored in water. It would appear it's being stored in a mouse. It would appear it can be stored in cell medium. Of course, we have no idea what the technology is behind this and what is possible to store it in and what it's not possible to store in. Ongoing research project. But before we get all the tingle, and Jerry goes marching off and says, hey, you can store healing in water, it turns out that healing has tremendous amounts of methodological complications. In particular, we have this resonant bonding. It's an article of faith out there, probably outside of this room, that physically separate things are independent of each other. And it turns out in the healing stuff, we take mice and we physically separate them. When we treat this, they don't seem to really be separate. And so we have the problem of even those parts of our experiment. This is going to come up with something called meaning fields, we think later. Even parts of our experiment, or mice or cell cultures or whatever, which are not treated but are part of our conceptual experiment, they also seem to get the treatment. And so we have incredible methodological problems. So, if I treat this cage of mice, and there's another cage of mice over here that's a control, and this group gets treated also, was it the water? Was it really the medium? Or was something else going on? So it turns out healing research is pretty difficult methodologically. So here's an example of an experiment. It's not really to scale, but we had more than 3 cages. We got cages here 100 meters apart. Away is a control not seen by any of the volunteer healers. We also had second controls that we didn't know, the healers didn't know even existed. And over here, we've got labs of the exact same mouse. Now, this is actually going to be a lead into a month, but if you believe in field effects, and this extends to a certain distance, anything presumably in the range of the field should be affected. But here's what happens. These other labs, independent, mice die on schedule. Same exact mouse model. Our mice get cured. Control mice get cured. This happens in 16 experiments in 6 different labs. And the control mice not known to the healers are also getting cured. So we need some model that, what's the influence and what's the extension of the influence and what ties it together? We're going to have to wait for a month. Bonds appear to be fluid. And an interesting area will be trying to figure out the laws of bonding and unbonding. Bonds can be broken. This is my all-time favorite experiment. This is to scale. These are 5 cages filled with mice on a single lab bench. And we have a control lab that's in the building. And we have a second control site trying to get these suckers to die, sent to another city. 3 of the healers are biologists. They don't believe in healing. They think healing is crazy, all that kind of stuff. 2 of the healers are non-biologists. They don't believe in healing. They think healing is crazy. The only difference is one are biologists, one aren't. We told the biologists that there's a control someplace in the building. Don't go look for them. Now, if you tell students not to look for them, okay, they said, and they peeked. Now, they didn't look for them, they just went there for 10 minutes. You know, so that in their heads, they didn't look for them. Now, here's what was happening. The control mice were dying on schedule, as predicted, as per history, and the biologists went to peek at them. And as soon as the biologists peeked, they weren't there, they swear, more than 10 minutes. The control mice started to go into the remission pattern. And they were eventually cured. Now, here's the interesting part. The biologist mice died. And they're treating them every day. And they died. And the non-biologist mice lived. And the control mice that was only seen by the biologist lived. A lot of head scratching goes on in this field, you know. Layered anomalies. This is a wonderful experiment. Now, here's the other thing. I'm not going to put this slide up, so I won't put it up here, because this is kind of a violation of every Ayakuk principle there is. But in this particular experiment, we had everybody take a cage home. You're not supposed to take mice home, particularly if you have cats. But you don't take a cage of mice home. But I wanted to see, could they do it at home? I was predicting, and again, Brenda, I yield, I'm almost always wrong. I was predicting, trying to find out, can healing be taught? If we got all the mice cages on a bench and there's any type of a field effect, if any one of the 5 healers could heal, they should all get healed. But what happens if they individually take them home and no one else sees them? If they take them home, everybody's cured. Even biologists can heal at home. But it turns out biologists can't heal in the lab. Now this goes back to the great Bernard Grad. I'm bowing. The granddaddy of all healing research. And Grad found at McGill in the 1950s and 60s, testing medical students, what is the effect of medical students who intend to heal? And it turns out they have an effect. They retard healing. His conclusion, If you're sick, don't go to anybody who's been near medical school. But you see the problem, methodological problems here. Was it the cell medium? Was it the cell transplantation? Was it the water? So I set up an experiment with Jerry. We haven't done it yet. I set up an experiment with Jerry. We got a blind for this. And what happens to the water and what's the information in the water? These are reasonably interesting questions. So, same question. Does it fit in the battery? Should it be a biological battery? Could it be stored in biology? Should it be a physical battery? Seems to be able to be stored in stuff. That's the question. So here are my partners in crime. First guy's in the back, John McMichael, Beech Tree Lab biologist. Dean Radin, who many of you know, so we're working on this. Dean has a weird tendency to walk around taking people's temperature. We don't know why that is. So you go, hi, Dean. He goes. Mike Turner, who's going after, I just saw Mike before. Mike's coming after us. And Sarah Beseme, who's going to be going at the tail end of this talk. And we're trying to figure out what's the best approach. So these are my partners in crime. And now, just thinking about this, if you take a biological approach to how do you store healing, we got problems. So what if healing could be stored, and what if we could make a vaccine? So the good news is we can make a vaccine. The bad news is, yeah, go try to use the vaccine out there. How'd you get the vaccine? Well, we call it the hocus pocus vaccine. You know, I mean, what do you do? So I'm trying to do an end run here and see if we can make a physical solution which would be more reproducible. Can we record it? Can we store it? And can we reproduce it by a physical storage? Even if we can make a vaccine. So here's the plot. Cotton. Using cotton. This goes also back to the great Bernard Grad. He discovered that cotton will store energy and store healing and store all that kind of stuff. And we have used this to some good use out there in the world. Apparently cotton is very good at this, and it turns out to be a healing potential. It's not a consequence. Cotton doesn't take intent. Cotton takes healing potential. It's the battery. And now if someone uses it, they can use it for X, they can use it for Y, they can use it for Z. It doesn't make any difference if everybody else knows about it. Cotton seems to be a donor. We don't know what else can do. So the question is, here's another complication. Can we do this? It seems to matter if there's a healing need. Different talk, but I think healing is brought upon by the healee, not the healer. And here's an example of interesting things that happen when there's a need. Mice, in my experiments, no matter who's doing the treatment, go to the left hand when they're sick. We spin the cage, they don't like that. When the cage stops spinning, you put your hands down and they'll try to move to this spot. Everyone in my system becomes left-handed. That's interesting in and of itself. But when the mice have a need, they go to the left hand. Once they're cured, there's no attraction to the left hand at all. So here's an example. These are nude mice. The cage has been spun, and they got tumors, and if we put this into a movie, you'd see them rotating around this spot. Doesn't make any difference the kind of cancer, doesn't even matter the kind of mice. This mouse has had its turn, it's going to walk its way around, and it would be a really interesting movie to shoot. So we take cotton, and the question is, do you have a need? So we've taken cotton and we've given cell cultures that don't have a need, they're just your basic cells, and we put cotton near the cells, and nothing happens. No change. But if the cells have a need, these are cancer cells, then we get genomic changes. So in other words, short version now, we're going to get more detail later, biology in need seems to be able to figure out whether cotton is charged or not. So we got your basic out of CVS cotton, and we got the cotton that's been applied hocus pocus to it. It looks the same. You put it under a microscope, it looks the same, but if a cell doesn't have a need, they can't tell the difference. But if a cell has a need, the cells can tell the difference in whether cotton is changed or not. There are genomic changes. Can gizmos, technical term, detect whether cotton is charged? And the answer is no. So far, Mike's going to talk about this in a couple of minutes, using different, but gizmos off the shelf and we put cotton near, and we do that, they don't seem to be able to detect it. And so a question, it's another question, I don't know the answer, maybe life needs to be the detector of healing. Maybe it has to be alive. Too early to conclude that, but so far it seems as if life has life in need, not just life in need has an easier time telling whether something has been charged or not. So we go back to, can we record the charging of cotton, play it back, and reproduce the cure? So we get Superman. This is at Dean's lab at IONS. We're not trying to be cool here. We're doing infrared stuff. We don't want to be blind is really what's going on. And Superman was watching over us. So we're in a Faraday cage of some heft. It's big and industrial strength and kind of thing. And inside we have all sorts of gizmos and a little setup here and a little setup here and setups here and different kinds of detectors. Actually 38 different kinds of detectors, all being fed into a supercomputer with how to be water processed because we got so many processors and we got all that kind of thing. So can we record, not healing, can we record the act of charging cotton? And here you have potential. You're not doing it for a particular end, you're doing it as potential. In the works here, sooner or later, we hope, we'll be looking at healing potential, charged cotton, on stem cells potential, and see what happens if potential meets potential. Don't know the answer to that yet. So can we tell the difference, or can we see, can we get something in the battery, and can that signal reproduce the cure, and does it matter whether the detector is alive or not? So we've got 51 different recording sessions in the Faraday cage, and we've so far, and Mike's going to give more on this in a little bit, but we, by objective measures of any worth, we're not getting a reliable healing versus control difference. He's going to talk about electromagnetic bursts, but people can feel the damn thing. I have it here. If anybody wants to listen to it later, we can figure out a time to listen to it. And very weird stuff happens when you play this recording. By very weird, I mean people have physiological reactions. Conditions disappear, weird stuff happens, I have no idea. Mike's going to analyze what's in it in a bit. But we put the stuff on, and people can feel it. Now, it's his turf, but there's not really much there. And I think, we don't know, the people who feel it in the place, here comes the extension, that they have a need. People who seem to have, this is only loose anecdote, heart problems, feel it in the chest. People who have back problems, feel it in the back. People who have knee problems, feel it in the knee. Just reporting. And so the question is, can we do that, and what happens if we play this stuff? And this, Sarah's going to now take us down a path of what happens if we play this stuff to cell culture.
So hello, everyone. I'm going to present you what we've been doing at Beech Labs. We did some genomic studies, so that's we want to measure any effect of these recordings, whether it's in vitro or in vivo. We are scientists and we want to prove everything. So this is our setup. We had to think really hard how to expose the cells to the recordings. We don't want to expose our stock cells to this recording, to this energy that it's storing it. So we set up a little incubator. We put some hold on, is it? Speakers inside the incubator. We've got a amplifier that's plugged to a normal computer that plays the recording and repeats. And so we tested 2 recordings to start. We have a third one that we're testing right now. And we play them sequentially to the cells with some controls with no sounds. And so this was the design of the experiment. First, I called it NS1, so it was a no sound control one. Then this is, so Bill called it the full spectrum recording, and I think Mike Turner will explain what that means, really. I can't tell you. Then the third exposure was what it was called scalar recording. It was a second recording. And to finish, we had another set of control cells that were exposed to no sound at all, no recording. This design was to compare what would be the genomics of these cells when they're exposed to the recording via no sound. So hopefully we can see a difference and get a first idea of what to look for in terms of mechanism of action. And also this control here was designed to, in case there's any remaining effect, or Bill called it the resonant, as he said. Because of one of the theory was also that this energy could stay in the room, and so you'll see that's the first question we really ask ourselves. And that shows you, so we use these genes that were identified in the cotton study. I didn't put the full name of the gene because it would be just confusing for us. So here on that graph, I compare, this is different time of exposure of the recording, so either half hour, 4 hour, a full day, or 2 days. And this is the level of expression of that gene. So in white, you see this was at the very beginning of the experiment. Then we played the recordings to the cells, and then we have another set of cells that were not exposed to the recording after the other cells were exposed. And you see a somewhat significant difference, which was surprising. These cells are supposed to have a very stable level of expression. You don't see that every time point here. For those who are familiar with this kind of graphs, these bars here tells us that there's not an actual difference. So we looked at all the other genes that were from that studies, and all these red boxes shows you that there was actually quite a number of difference between these two controls. So what does that tell us? That the recording, even after, seems to have an effect on the expression of certain genes after the exposure, which correlates to Bill's resonant bonding idea. Does the energy stay in the room? Is it stored in the incubator? Is it stored somewhere? And can an energy cleaner fix it? So we worked with Yuri, who has a clean sweep spray that can clean the energy of the room. You see me a little talking without knowing very much. I'm a very dubitative person, so I want to try it, see if it works, and then I'll tell you, oh yes, that cleans the energy. Before that, I won't tell you it does. So why not? Let's try it. It's in progress. So while we're going to design we are designing the same type of experiment. We'll spray the clean sweep, and we'll see if we still see that difference between the controls. And once we have an answer to this question, it'll probably lead to more questions. And I'll see you next year, I think. The second question we were asking ourselves, well, okay, we have this recording of healing activity. Does that do anything of the genes that are involved in cancer? Well, so now we're comparing in black and gray. You see that that's the level, the gene level for cells that are exposed to these recordings. And you see these arrows shows you that for these genes, not all of them, there is a difference. When the recording is played to the cells, the gene is usually upregulated, means there's more expression of that gene. What does that mean in terms of biology? We don't really know now, but what we know is that there is an effect. And this experiment allows us to have a first, give us a direction to look into for future experiment. So we had this result. These were We did that at different time point, as I was showing you, so even more arrows that shows a difference. So next step is what we want to see. Okay, we proved that some genes are they can be. There's an effect of this recording. So we have a few preliminary genes that we identify. What if we can probably identify more pathways or more genes that could be modified? So we enlarged our panel of cancers, and we looked at genes that were involved in cancer pathways, because those are cancer cells. With the results that were seen in mice, and this idea of there might be an immune component to the healing process, we also were interested in genes involved in immunology and inflammation. So we looked a bunch of genes. In each pathway was a little over 80 genes, so it's more than 160 genes at multiple time points, so that was a pretty extensive genomic study. That's just an example. I'm not going to explain, and I'm not going to go through every single bar here. That's just one example of the amount of data we accumulated. And in summary, so these here are genes that we identify in that first really screening that seems to respond to the recording that we were playing. What's interesting is some of these genes are involved in apoptosis. I don't know if any of you is familiar with that pathway. It's programmed cell death, so it's very, very important in a lot of disease. This pathway here, telomere length, is also very important in aging or age-related disease. So this is a first step toward possible understanding of what's going on, to maybe explain the reaction of people who are sensitive to this recording, and if we can explain some sort of mechanism, a biological mechanism, maybe we can get to something we can actually provide and help people to heal. So we're continuing gathering data on now we're going to look at this, it was 10 genes, a little closer. Also, we have a new recording that was recording in a different way, not in a Faraday cage, but not in a cage, in a studio. It was a little improvement in working conditions for Bill. And we hopefully will begin animal studies if we get some more interesting results in vitro. Okay, so we thank the Emerald Gate Charitable Trust for their support. And this is it. That was Bill and me.
Thank you very much. Yes, you mentioned that you've done 84 different tests, and I'm seeing error bars. Are we correcting for multiple tests in terms of our probabilities on these experiments?
You're the one who ran it.
But you're the statistician.
No, I haven't, no. Yeah, I don't.
Not yet.
They're running, again, the standard stuff that they do, but I haven't come in as a statistical over-your-shoulder looker yet. Yeah. If we're wrong, everybody else in biology is wrong. So, but that's not an answer to your question.
You mentioned that this was a transfer of information rather than energy. Why do you say that?
That's a long answer, Sam. Just very briefly, for example, if we're talking about traditional kinds of energy, and I'm talking to you and I'm backing off, then I'll trail off and get quiet. But healing, we do it at different distances and such. There doesn't seem to be any difference whatsoever. You do a mouse experiment at 2 inches, and you repeat it at 2,000 miles. It seems to be the same. There's no diminishing. It's a complicated answer. It also has to do with the kind of cancer you treat. It turns out the more aggressive a cancer is, the easier it is to fix. So given similar kinds of doses to a very slow-growing cancer, giving the same dose to a fast-growing cancer, the fast-growing cancer is going to respond much quicker. The energy is, the information would be the same. The energy is in the system itself. It's in the disease. So very aggressive cancers leave very quickly. I mean, I can keep going, but you can see it becomes a pretty interesting problem to try to untangle. Steve, you're in charge, so their hands up.
Yeah, we really are out of time, but if you'd step to the mic real quickly, we can maybe take one more quick one
I
here.
just want to know, does need have an intelligence?
My guess is yes. That's my guess, but that's not worth a penny. I'm running through some studies I could do based on your question. I don't know the answer to it, but my speculation would be yes. It would seem that way, but I can't say I did this, therefore I'm concluding it.